Today we're going to talk about the endocrine system. Hi, I'm Larry Johnson. I am a professor from Texas A&M University, and today we're going to talk about the diversity of organs in the endocrine system. This is part one. Here we can see the endocrine system components. The pituitary is at the top. Lower left is the pituitary. You can see some of the cells that are located therein. The adrenal is in the middle. And the far right, we have the thyroid gland. So there's a diversity of different types of organs, and those have functions. And that's what we're supposed to do today, is to gain a greater appreciation for the diversity of functions of the endocrine system, and to recognize the different organs and unique features of organs and cells that make up the endocrine system. If this information is useful to you, please share it with your colleagues. Thank you. Today we're going to talk about the endocrine system. Hi, I'm Larry Johnson. I am a professor at Texas A&M University. And today we're going to talk about the endocrine system, which is composed of several organs and regulations and negative feedback of the process. We want to gain a greater appreciation for the diversity of functions of endocrine system and to recognize the different organs, unique features of organs and cells that make the endocrine system. Here we can see the pituitary gland, how it interacts with a host of other tissues, but it itself was interacted with by the hypothalamus. So the function of endocrine system is to regulate metabolism, growth, development, tissue function, sexual function, reproduction, sleep, mood, among other things. And here we can see some different organs. There's pituitary gland, there's adrenal, this is the ovary, and this is the pancreas that we'll be talking about next time. Now, the first scientific endocrine study was in the testes of roosters, studying observable effects of testosterone on the secondary sex characteristic, that is, the wattle and the comb growth. If you transplanted testes back into the abdominal cavity, it prevented the reduction in the size of wattle and comb growth in castrated roosters. If you remove the testes from a rooster, his wattle and comb growth regresses and looks more like a hen. But if you transplant the testes into the abdominal cavity, severing anatomical connections, putting in the abdominal cavity, and the cells grow in there, and they produce hormone, which maintains the secondary sex characteristics. And toward the second part of the endocrine study, we'll talk about transplanting testes into rats to study the effects of hormones as well as on Sertoli cell number. Endocrine study, we see the testes here, we see the adrenal, both in the young and the adult. Now here we see two boys. This guy is 9 and this guy is 10. And you can see that his growth is stunted because he hasn't had sufficient amount of growth hormone. And in fact, if you don't grow by 2.5 inches a year between years 3 and 9, it's a red flag for something going on during that point in time, and they will give you therapy. Now, a dwarf was used for ET. It was a robot, was most of the acting, but sometimes a dwarf was used by there. Or a giant is another endocrine effect. Here you can see a giant. Here this guy was 8 feet 5 inches tall. Also, Bo was from a tribe that has the tallest people in the world. And you can see it'd be no problem for him to dunk the ball. But I think some guys would shoot right between his hands. I'm not sure. Introduction. We want to look at overview of endocrine system, define the endocrine system, gland secretions of hormones, and look at physiologic levels. Now, physiologic level of glucose is 10 to the minus 2 molar. So it's fairly concentrated. However, steroid is to the 10 to the minus 9 molar. And the peptide is even more minute. So these are different concentrations, so a minute amount of hormone will have a great effect, as we can see on these two individuals. In fact, if you have 10 to the minus 13 molar of growth hormone, you become a dwarf. But if you have more, you become a giant. So the growth hormone is important in stature of the individual. And we'll talk about the control of the endocrine system. Now here we can see that endocrine glands, they have no ducts, highly vascularized, rich vascular supply, characteristic of endocrine glands because they have to secrete their hormones in the connective tissue and get into the bloodstream, is what they do. Some of them secrete the hormone directly into a lymphatic, for example. Secretions are stored in granules, maybe, or they may be stored in extracellular space, like the thyroid, or maybe no storage at all, like steroids, which can walk right through a membrane. We have anterior pituitary, posterior pituitary, and these come from different locations. The thyroid, we can talk about the parathyroid, is cords, adrenal, the pineal gland, and the pancreas are some of the things that we'll talk about. The pancreas have both endocrine as well as exocrine function. Pancreatic juices are produced. So the endocrine means internal secretions without ducts. Mostly it comes from the endoderm, but there is some from the exoderm, and we'll talk about it. Hormones equal to arouse or to set in motion. Internal secretion, hormone to set in motion. So the origin of different epithelia comes from a different layers, from the ectoderm, endoderm, and mesoderm. Most of the endocrine glands come from the endoderm. However, the pituitary comes from the ectoderm, as we'll see a little later on. So adenohypophysis comes from the ectoderm. Endoderm is where you get most other endocrine organs are produced. And they may be originally connected by a duct, but then that duct regresses and it's loose in the connective tissue. It's where we see the endocrine cells. So here we see the thyroid. There's the adrenal, or some of the endocrine organs that come from the endoderm. And here we can see release of neurons, and you can see some of them have direct release where they release their transmitters onto muscle cells or other cells. And in the case of adrenal, it does the same thing. It releases it. Epinephrine is released due to neurological stimulation to the cells that produce it. However, indirect effect of target organs is what we have in the endocrine system, where you have pituitary hormones that stimulate various glands. Or also you can have pituitary hormones that affect other glands. In this case, the hormones are actually produced by nerve cells, where here it's produced by epithelial cells. So as I mentioned, a paracrine is when one cell signals a cell adjacent to it. Also, you can have a juxtacrine, where one cell stimulates another cell by direct contact with that cell when we have that during the embryologic development. There's neurotransmitters that are used for one cell, a nerve cell, to interact with the target cell. But in an endocrine cell, you got a cell located one place, secretes hormones that gets into the bloodstream that affects the target cell at some distance. And so here we can see synapses. You can have synapses on different cells, different kinds, but in the case of endocrine, you interact with different cells by different hormones, and these hormones have specific receptors on the responsive cells. So cell A produces a hormone that interacts with target A, B, C, C. And so some of the receptors on the cells are located on the surface, and some second messenger comes in to stimulate the cell. Others are actually located in the cytosol or maybe in the nucleus itself, and that would be a carrier protein would carry it into the cell. Now, of the second messenger one, we have a hormone with a receptor. It calls cyclic AMP to be produced. And here's a host of hormones that are produced by these various tissues and cause these responses, and it's due to the cyclic AMP being increased in these cells. In fact, if you stimulate the cell by increasing cyclic AMP, it will cause that same effect to occur. So here's actions of cyclic AMP. You see in the liver, increases different enzymatic activity. In kidney, it increases renin, nerve, increase acetylcholine release. And so these are cyclic AMP, second messenger effect, where you have like a protein hormone that has a receptor for the outside of the cell. In the kidney, increase phosphorurea, increase renin, for example. Now, the pituitary gland, or the hypophysis, as we can see in through there, and it's coming right there, just as located right below the hypothalamus, produces 9 hormones. It has a reciprocal relationship with the other endocrine organs. Both it stimulates organs and they stimulate it. It's a neurological vascular connections, and we'll talk about the vascular connections that go here, and also nervous connections that are attached to the hypothalamus. And it's located in a key place for a neuroendocrine system to develop. So by the brain telling the hypothalamus to secrete releasing hormones, stimulate pituitary to stimulate the various organs. So you have the brain regulating the pituitary function. And here we can see the pituitary is divided in 2 groups. You have the neurohypophysis, which is the pars distalis, the pars tuberalis, and the pars intermedia right in through there. Then you have the neurohypophysis, as we see here in blue, and so that has the pars nervosa, the infundibulum, and the median eminence that's located in through here. So the different parts of it and how it gets the vascular supply is different too. So we can see that again, the adenohypophysis, pars intermedia, pars distalis, pars tuberalis. And we can see those here. The pars distalis is very cellular, that we can see. Pars intermedia is cellular as well. And then the pars nervosa is more nerve-like tissue with pituitary inside there. Here we see the pars intermedia, pars distalis, very cellular as you can see, and a lot of connective or nervous tissue in the pars nervosa. Again, these are the different areas, the infundibulum stalk, and this is the pars nervosa, pars intermedia, and pars distalis. This happens to have a little cancer in him, but it's useful for us to see. So we see the pituitary, the pars distalis, has 3 groups of cells. It has a chromophobes that doesn't like stain. It doesn't like stain. And so what we see here is a nucleus. We don't see the cytoplasm staining because it doesn't like stain. And then we have acidophils, and they are the one that have red cytoplasm, or the basophils have blue cytoplasm. So there's the nucleus and there's the cytoplasm that we can see there. Now in the pars nervosa, we have pituitary, which are support cells, and then the cell bodies for these guys are actually in the hypothalamus. So we don't see cell bodies, but we do see swollen areas where hormone is concentrated, and those are herring bodies. You can see one right there, and we'll see some larger ones in a minute. Now, the pituitary has a binary origin. It comes from the roof of the mouth, Bronchus pouch, as we see, and then that connection with the surface of the mouth is eliminated, and so you have the pars distalis, and so this is epithelial type cells from the ectoderm or the oral cavity. In contrast, you have the pars nervosa, the neurohypophysis, coming in through there, which is how the outgrowth of the brain. So you got from the oral cavity going up, or the brain coming down to be a origin is two sources of cells, a binary origin. And these cells have neurons that are located here, and those neurons run down all the way down and store their hormones in the pars nervosa. Is what we see. So again, we see from the roof of the mouth down from the brain, it comes from the ectoderm. Is where we see this part. A brain comes from ectoderm anyhow, but this comes from the brain as part of it. Again, we can see the same type thing coming up from the pouch, coming down from the brain, and then ultimately you have intermedia in the middle. And we can see the pituitary, and here we can see the adrenal developing right here, a little higher mag of that, from the adrenal developing at that point in time. So as I mentioned, neuron cell bodies located up there, some of which apparently synapse, releasing their hormones in there. So they are connected vascularly as well as connective tissue. We have chromophobes that don't like stain, acidophils, and basophils is what we have. And here we can see those again with this particular stain. You can see the acidophils kind of brown, and the basophils are otherwise, and the chromophobes are those here where you can't see much cytoplasm. Again, you can see with this slide, you can see the acidophils, you can see the basophils, and the chromophobes didn't like stain. And the acidophils and basophils, they produce hormones that interact with different organs. So acidophils produce prolactin for the mammary gland to produce milk secretion, as well as growth hormone, and also antidiuretic hormone comes in through there. The other ones that go to these organs goes to the adrenal cortex, the thymus, the testis, the ovary. Those are the basophils that produce hormones for those. And here we can see the staining for the different types of cells. Pars distalis, you can see it's rich. This is one of somatotrophin cells. It has these granules, smaller granules with other types of cells, and mammiotrophs here. So different cells are all together there. Somatotrophs again. This is gonadotroph. It got varying size of granules, and so just different one mammiotrophs. And the pars distalis, if you look at the blood supply, which is shown here in black, you see it's very vascular. It's very vascular because the endocrine system, the blood system, is how the hormones get to stimulate these guys, and also how their products get back to their target organs is through the bloodstream. So the variation in microvasculature, as we talked about before, the common is arterial, capillary, venule. That's your common thing. In a portal vessel, you have two capillary networks, 1, 2, and you had a portal vessel in between. Okay, and that portal vessel could be a venous one, like hepatic portal system, or it could be arterial one, or portal arteriole, like here. The afferent and efferent arterioles are a portal arteriole, as opposed to a portal vein that we see. And so the portal system functions to create a local change in blood composition. So in the first case, the first capillary network, it modifies blood composition. It puts a hormone in, it distributes hormone. The second one is how they benefit for that local concentration. As I mentioned to you, the hormones are low on concentrations, 10 to the minus 9 or 10 to the minus 12 molar. That is real low. And so in order for you to stimulate the cells, in this case, you need to have from the hypothalamus to the pituitary, you need to have a local control system so you don't dilute out the hormones to stimulate them. And that's what you have, a local portal system. It's for local connection. First capillary network modifies the blood, and second capillary network benefits from that modification. And here we can see it. So the first capillary network is located right here in the hypothalamus. And the second capillary, this is a portal arteriole here, second capillary network is in the pars distalis. So you get releasing hormones that stimulate these hormones to produce ACTH or the various components that are being produced in the basal fields. Capillary network, portal system, and then the second capillary network. And here we can see they're releasing hormones in these cells, and they're so close to the endothelium. So hormone is released in the connective tissue, seeps through the endothelium. That's in the first capillary network, and then in the second capillary network is where the hormones are released to stimulate eosinophils and the basophils. Now, in addition to having a portal system of pars distalis, you have the nervous system comes in through, and a different blood supply comes in. You don't need a portal system here. You do need it over there. So there's no portal system on the pars nervosa, as we can see. And so if we look at the hormones that interact, the eosinophils and the basophils, you can see there's a host of organs. And these organs produce hormones too. FSH stimulates ovaries to produce estrogen. Estrogen feeds back negatively. So we have negative feedback. So as you stimulate the thyroid, it produces a thyroglobulin, which is converted to thyroid hormone, and then that inhibits further TSH secretion. And that happens to ACTH, LH, FSH. You have a negative feedback system to prevent overproduction of these. And from the eosinophils, here we got the growth hormone for your muscle, fat cells, and prolactin being produced as well. And so here we can see the pars nervosa, pars distalis having a portal system, pars nervosa having only a capillary network, a typical system that occurs. And the pars nervosa, as we mentioned, there is the neurohypophysis of pars nervosa, infundibulum stalk, and a median eminence, as we talked about before. So we'll talk about the pituitary, which are the nerve cells. Herring bodies is the storage, and neurosecreted granules is what's being released, and secretion by nerves up at the top. What we're going to talk about. So here's the infundibulum stalk inside the tuberalis that we can see. Pars nervosa. So we're talking about these. A nerve cell bodies are located in the hypothalamus, and they discharge all along and through here into this clear region, as you can see. Pars intermedia, pars distalis. Again, we can see the herring bodies are these swollen portions of the axon. You can see it right here, granular. And so we're talking about this portion in through here. And what we see there, as the nerves come in through there, there's swollen portions along its length. And that's the herring bodies. We can see them. There's a herring body there, 1, 2, 3, full herring bodies. You can see them. They have a membrane around it and granules inside there because they're actually inside the axon of the cell. And so these produce different things. They stimulate their oxytocin for contraction, oxytocin for milk, mammary gland milk ejection, and antidiuretic hormones. So these are some of the hormones produced by these cells in the hypothalamus that store in herring bodies here until the hormones are needed. Oxytocin, ADH, that's there. And here we can see the herring bodies, many herring bodies that we can see there. And remember again, they are the swollen part of the axons along the way. And so you have the action potential comes down through there. You have calcium influx and exocytosis of the transmitters onto the connective tissue, which is right next to a capillary. And so the hormones are going to be dumped into here. The oxytocin, ADH is going to be dumped into there. And the herring bodies. You can see the herring bodies are really swollen part of axon storage areas. And so that puts a pituitary together. You got part that you have neurological cell bodies that are located in there, so those neurons discharge oxytocin. And then you have these other cells that produce growth hormone or prolactin, acetyphils, or the basophils produce other hormones. And this having a portal system, this having no portal system that we have. Because you have the neurological connections, you don't need to bring hormones. The hormones are going to come down the axons in the case of the pars nervosa. Here we see ADH, normal amount of, this is salt-treated depletion of antidiuretic hormone. So if you give salt depletion, you're going to discharge your ADH in the pars nervosa. And here we see normal amount. This is pars nervosa right here, and you have hypothalamic diabetes that we have. Again, you have depletion of the antidiuretic hormone. And so here we see where a little calf is drinking, or a baby, and that sends a message up to the brain. It calls the hypothalamus cells, those neurons, to secrete their hormone, oxytocin, and then it comes from neurohypophysis. Oxytocin comes down and stimulates the cells on the mammary gland to cause milk ejection to occur. Thyroid stimulating hormone is another hormone. Its significance is to regulate metabolism. And the hormone is actually stored, a precursor to the hormone, the thyroglobulin, is stored extracellular. In order to keep it corralled, it has to be in a follicle. A follicle basically has no outlet. Anything that goes in and out has to go through cells, and that's what we're talking about. So this is a colloid. A capillary is a rich vascular supply around, and the follicular cells. So these are follicular cells that have tight junctions between one another that makes a sealed follicle for the thyroglobulin to occur. They're follicular cells, and there's connective tissue and blood vessels that we can see. So here we can see the cells. These are follicular cells, columnar cells, and they discharge thyroglobulin, and they take thyroglobulin in and produce T3 and T4, as we can see. So here we see the follicles. Follicle, again, is a tight junction cavity to hold the extracellular stored precursor hormone that we see. In addition to the follicular cells, in between there we have the parafollicular cells. The parafollicular cells, and they are important in produce of calcitonin. And so here we see the follicular cells along the way, and the capillaries, a rich vascular supply. So these are the follicular cells which produce the colloid And here we can see from the brain you got coming in a nerve cell, and then you got the hypothalamus, you got production of precursor hormones, of stimulating hormones to come to the anterior pituitary, the pars distalis, stimulate these cells to produce thyroid hormone, stimulating hormone, stimulating hormone, produces thyroid hormone that goes to the target cells. So here we see the thyroid here, the parathyroid there, and this is iodinated thyroid. If you take iodine, radioactive iodine, and you show it concentrates on the thyroid. Here we can see the radioactive iodine has concentrated on the thyroid cells. The thyroid gland is rich vascular supply. You can see a mesh of blood vessels around each of the follicles. Here we see follicles and rich vascular supply there. So in terms of release of cells, epithelial cells, there's 4 basic ways. You have American, which is just plain old exocytosis, and that's what happens to proteins. You have Apican, where you have the loss of part of the membrane, and this is a case of lipid excretion in milk. Then you have the Holocrine, where you got the whole cell secreted. That's the sebaceous gland. Then there's another kind, there's the cytokine secretion, where you have myelin granules transferred from the melanocytes to the keratinocytes. So you got a product from one cell which picked up by the other cell. The darkness of the skin is due to the myelin granules in the keratinocytes, even though they were produced by the melanocytes. And here we can see the American secretion, where you have thyroglobulin being produced by these cells, and talking these thyroglobulins in through here, which then is phagocytized, broken down, and produced upon TSH stimulation and iodine. They produce thyroid hormone. Thyroid hormone stored in the colloid, as we see in through there, and these are the follicular cells. Thyroid, the production of thyroglobulin does not require iodine, but to break it down does require iodine. So if you've got an iodine deficiency, you'll produce more of these, more colloid as a consequence of that, and you don't break it down and such that you have a goiter. So goiter is accumulation of thyroglobulin and iodine deficiency is what it is. Graves' disease is hyperthyroidism, IgG, immunoglobulin, long-acting thyroid stimulator. So it thinks this IgG immunoglobulin is actually thyroid stimulating hormone, is what happens in that case. Thyroid stimulating hormone, as you see, comes from the hypothalamus down to the pituitary, goes to the thyroid gland. T3, 3 to 4 comes back there, has a negative effect on both the pars distalis as well as the releasing hormones being produced to stimulate the pars distalis from the hypothalamus. The parafollicular cells, as you see, these are follicular cells, but then there's parafollicular cells located in through there. Here we can see another one here right between the follicles, another one right there, and they produce calcitonin. So in terms of calcium regulation, the parathyroid hormone stimulates osteoclast production, but calcitonin removes the osteoclast ruffled border. If you see an osteoclast ruffled border right here, which makes a microenvironment to allow it to digest bone, if you pull that ruffled border up, you lose your microenvironment, and that's what calcitonin does. So endocrine secretions, they could be stored in granules, as you see there. They could be stored extracellular, as in the case of the thyroid, or immediate release, as in the case of the adrenal. Protein in cell, thyroglobulin outside the cell, or steroid to pass right through the cell. The parafollicular gland, the parathyroid gland, you can see it's 4 of these parathyroid glands. It's important. It has parathyroid hormone, regulates calcium, and you have secretion. You have chief cells and oxyphils. We don't know the function of oxyphils, but the chief cell is the main one that produces parathyroid hormone. And so here we see the parathyroid hormone in the thyroid. This is that we can see. It's these dark cells in through there. They have the chief cells and the oxyphils. Oxyphils are big cells, dark nucleus. We don't know what to do, but they are pretty in there. And then these are the chief cells that produce the parathyroid hormone. Oxyphils, chief cells that produce it. Again, the parathyroid hormone is produced by the parathyroid gland, which is very vascular, as you can see with these dark blood vessels in through there. The parathyroid glands produce parathyroid hormone, and it acts on bones, both osteocytes and osteoclasts. It also works on the kidney to increase reabsorption in the distal tubule. It works on the intestines to increase calcium absorption, to maintain a tight control of given concentrations of calcium, which is 8 to 10 milligrams per 100 mils. Calcium necessary for muscle contraction, glandular secretion, blood coagulation, key enzymes for inflammatory metabolism. Removal of the gland results in violent spasmodic contraction, tetany, and ultimately death. You need your parathyroid glands for the parathyroid hormones. So the parathyroid hormone acts on bone. It acts on the osteocyte, osteolysis, that is, mobilize calcium. Osteoclasts increase calcium concentrations in minutes. This is a quick thing. Also, osteoclastic bone reabsorption. It caused by prolonged hypocalcemia. You get a cascade of additional osteoclasts. So you have additional osteoclasts differentiate and start to break down bone. So one is a long-acting and one is a quick-acting response to parathyroid hormone by the two different bone cells. So here we see the parathyroid gland. These are the chief cells and the oxyphils. These are the ones that produce the parathyroid hormone. So we talked about it. Parathyroid hormone has a counterpart, calcitonin. One stimulates absorption of bone, and one prevents absorption of bone. The calcitonin removes the ruffled membrane on the osteoclast. Osteoporosis due to hyperparathyroidism is one of the problems that we have. So we talked about the pituitary gland, the adrenal hypophysis and its components, and the neurohypophysis and its components. We talked about how the endocrine system, it has cells in one location. Different cells produce different hormones, and that corresponds through to the receptors that are on unique receptors that are on different cells. So cell A reacts with cell B, cell A, cell B reacts to target cell B. Cell C's hormone interacts with receptor of target cell C. So hormones are produced to mediate the response on target cells at a different location. And there's a host of different cells, hormones that they produce, and physiologic responses that occur in the body. Next time, we'll continue with the endocrine system, and we'll talk about the adrenals. Is one of the glands that we'll talk about, and the testis and ovary somewhat. And also, we'll talk about some experimentation using the endocrine system is what we're going to talk about. And here we can see this is a number of Sertoli cells with various hormonal treatment that we can see. We want to thank the various books for illustrations that were used in this presentation. Thank Body Worlds for their images. These books produce these drawings and diagrams. I'm not the source of those. Those are from these books, and we thank these books for them. This is the Library of Congress. This is the outside. This is inside the Library of Congress, and we can see two people, a young guy studying and an older guy with wisdom. That we can see both of these individuals. Very lovely hall inside there. If this information was useful, please share that with your colleagues. Thank you.